Support Microbe TV. It's time for our annual fundraising drive. We depend on your support to produce high-quality science videos and podcasts, and now is the time to help us. If our programs have appealed to your science interests, or if they have helped you in some way, please record a brief audio or video with your phone and tell us about it. Let us know how we empower your inner scientist. We'll use it for our fundraising efforts. Send it to incubator@microbe.tv. And for more information on how you can give us your support, go to microbe.tv/contribute. This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV. This Week in Virology, episode 1181, recorded on December 27, 2024. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from College Station, Texas, Jolene Ramsey.
Howdy. Good to be here.
Gosh, we have two Texans here today. Also joining us from Austin, Texas, Rich Condit.
Howdy. Speaking of Texas, how are y'all?
Okay, it's our last TWiV for 2024, right?
Yeah.
Next one
It's,
will be
by the way,
next
it's
year.
81 degrees. And you can tell, because Tinkerbell is here, that it's sunny out. Tinkerbell only visits when the sun's out.
It's sunny here too, Rich, but it's actually only 71 and
Wow.
22 Celsius. Yeah.
All right, Jolene. So is your office cold? That's why you wear a shawl, or you just like the style?
I am just always cold. You know, growing up in Spanish, my grandma would call me friolenta because I'm just always cold, and I've been known to have cold hands, cold feet. So I moved to Texas to escape the cold. And I love it. The Texas winter is my style.
I'm wearing a sweater here at the incubator, and there's a heater here, and for most people it would be fine, but I'm too chilly. I bought a space heater, but it keeps blowing the circuit breaker, so I can't use it.
Oh, no.
Great, the space heaters that blow circuit breakers. If you like these programs on science, and let me just tell you, this is science. We give you science by scientists. These are people trained to do experiments and interpret them and plan new ones. So we're all trained to, what should I say, look at facts, science facts that emerge from experiments. You run a piece of DNA in a gel, you know where it migrates, the size. That's a fact. It can't be changed by naysayers. So we talk about that here. And I think we need this more and more in the coming years. So if you like what we do, please support us. We need your support. We're a nonprofit entity, Microbe TV. We don't do ads. The only way we can run is by your donations. So go to microbe.tv/contribute and help us out. I was talking to someone yesterday. Said, I buy this coffee at Starbucks every day. It's $8. $8 a day for a cup of coffee. Wow. You know, you could give us a couple bucks a month. It would help. It's not even going to impact your coffee intake. Isn't that ridiculous? $8 a month for coffee? Oh my gosh.
$8 a day. Yes,
sorry,
that's ridiculous.
$8 a day. Totally crazy. Microbe.tv/contribute. Release the viral immunologist in you. The Rosenfeld Laboratory and the Division of Viral Products at CBER FDA is looking for a postdoc staff scientist to continue the lab's research, understanding immune responses elicited during enterovirus infection. If you're an immunologist, microbiologist, or virologist wanting to learn more viral immunology and vaccinology, email amy.rosenfeld@fda.hhs.gov for more information. And ASV 2025 will be held in Montreal at the Palais de Congrès de Montreal, July 14th to 17th. It's Montreal for a lot of people, right? But I'm just trying to be proper. I took 10,000 years of French in school. I might as well use it. July 14th through the 17th. I try and speak French and it just degrades into Italian because Italian is in my genes. I learned Italian when I was a baby.
Okay,
up until
Vincent, Italian is not in your genes. Someone tells me that. They say Spanish is in your genes. It's not in my genes.
I know it's not
and
my genes,
Italian is not
but
in your genes
I
either.
learned it. Well, both my parents are Italian. I learned it when I was 0 to 5 years old, which is a great time to learn a language. And it is in my head permanently. I go to Italy, and within a couple of months, I would be fluent if I could spend a couple of months in Italy. And so if I try and speak French, it degrades inevitably into Italian. So anyway, July 14th through 17th, 2025, website asv.org/ASV2025. Abstracts are due February 3rd, which is less than a month from when you hear this. Travel award applications are due February 5th, so now would be a good time to renew your membership or join ASV. You have to be a member for travel awards, ASV cares, and on-site child care. And ASV members who are no longer trainees get discounted registration. Just use a search engine for ASV membership. Rich, you don't have lifetime membership, right?
I do.
Yeah, it's great, isn't it? We don't
Yeah.
have to worry about it.
And it was, partly it's just sort of my retirement gift to ASV. And partly I don't have to worry about it.
Yeah, I did it last year because I was tired of worrying. I was like so late. I apparently missed many years.
I mean, if you're an early career virologist and you anticipate being a virologist for the rest of your life, should you just become a lifetime member?
Sure.
Yeah, the problem is it's expensive as a young virologist, right? But that's what makes the most sense. If you're going to do virology the rest of your career, as opposed to Rich and I, we're at the end of our careers and we buy lifetime membership. That's stupid. It is downright stupid and not reflective of our skills as virologists.
Well, I contemplated doing it much earlier on. And then it turned out that my department paid for my my memberships.
No.
So
nice.
I decided, what the heck, I'll do that.
Why would you do that? Yeah. I'm sure they don't do it anymore, right?
No.
Yeah.
That's why
in general.
one of the reasons I became
I mean, once
a
you're
member.
a lifetime member, you're still going to be, asked and possibly feel like you can contribute to the other funds that the society has.
Mm-hmm.
It just means you don't have to worry about paying your membership dues.
It's a pain in the neck every year. I mean, it's not really. It's just that you forget, and then it becomes a pain. When you forget and people start bugging you, then it becomes a pain. Okay, asv.org/ASV2025. See you there. I have one news item, but this is very sad. This I learned from Daniel yesterday. This is a CNN article, but there are many others. Bird flu kills more than half the big cats at a Washington sanctuary. I mean, these are beautiful cats, right? Of all kinds. 20 out of 30 something, almost more than half of their animals.
African servals, bobcats, cougars, Canada lynxes, and an Amur Bengal tiger mix.
Yeah, it's very, very. I saw some pictures. It's just very majestic, and it was too bad that there was a tiger in there too. Wow. So these are really big cats. So they got H5N1. They don't know how they got it. It's so really unfortunate. Maybe someone brought it into the sanctuary. Who knows?
Well, they say also that there's they are in the path for the flight pattern for migratory birds. So maybe it was a wild bird dropping exposure.
I wasn't aware that it was that lethal in cats.
Yeah, in cats in particular, it's bad. Yeah.
By the way, I don't know if you were aware of the fact that I cut out there for 30 seconds or so again. It looks like this is going to happen. And I don't know what the problem is.
Maybe the recording may be fine. So nobody will notice.
Well, yeah, when it happens, you guys just freeze up and I can't hear you.
Oh, sorry.
We'll see what happens.
Anyway, I thought this was very sad because they're beautiful, just beautiful animals, right? And some of them are relatively rare, so you don't want to lose them. It's too bad. So I feel bad about that. I told Daniel, Daniel always says, no one is safe until everyone is safe. I said, we should include animals, non-human animals,
Yep.
We should protect them as well. Okay, I have a snippet for you, and Jolene will do the paper. And this is a Nature Communications article. Origin and cross-species transmission of bat coronaviruses in China. And the two first authors are Alice Latine and Ben Hu. And then the senior author is Peter Daszak. Also here are Jonathan Epstein, Linfa Wang, Zhongli Shi. And they're from the EcoHealth Alliance, the organization here in New York City, the Wuhan Institute of Virology, the Guangdong Academy of Sciences, the Duke National University of Singapore Medical School. So I have to give you a summary right up front. So what's the research question? The research question is, what is the macroevolution, cross-species transmission, and dispersal pattern of coronaviruses in bats in China? What is macroevolution? Macroevolution, I swear I had to look it up. I didn't know what it meant. This is macro and microevolution. With respect to this topic, macroevolution is the evolution of the viruses in multiple species. Micro would be in one species, okay? That's the way I look at it. So
that's helpful.
They're looking at multiple species here of bats. Okay, so that's one question. How do they try to answer it? Well, they've been sampling bats throughout China for years, and so have many other people. And so those sequences are available. So they took the coronavirus genome sequence, the kind of bat, different species, and where it's located in China, and use that information. They did computational approaches to try and get answers to the questions. How do they spread in China? Do they infect multiple bats and so forth? What does it mean? What does it mean, jelly bean? Well, what it means is that coronaviruses can infect many different kinds of bats. They can spread all around China, but there are epicenters for certain coronaviruses where they emerge, and certain bats are major hosts. And so if you need, at the end of the paper, they suggest, here's where we should do our surveillance. We shouldn't just survey every bat into China, right? That's no good. You have to be focused. And this tells us that this part of the country is where we should be looking. All right, so that's the overall thing. So as you know, all the coronaviruses, the human ones, are zoonotic. They came from animals originally. And many non-human animals have zoonotic coronaviruses as well. SARS-CoV, right, came from horseshoe bats. So horseshoe bats are a major player here. Rhinolophus genus, Rhinolophus. Those are horseshoe bats. They're the natural reservoirs of SARS-CoV, and probably where SARS-CoV originated way back in 2002. Swine acute diarrhea syndrome, SADS coronavirus. It's great. Pigs get diarrhea. It's fatal. Killed over 25,000 pigs in an outbreak a few years ago. Came from a Rhinolophus bat. It's a coronavirus. SARS-CoV-2, of course, most likely came from horseshoe bats. Why do we know that? Because very similar genomes have been isolated from horseshoe bats. Now, I say very similar, 96% identity. And by the way, someone wrote us a letter saying we should use identity, not homology. You can't say percent homology, right?
That's right.
You have to say percent identity, either amino acid or nucleotide. I always slip into it. I make the mistake all the time, but it's absolutely right. So 96%, in this case, nucleotide similarity. That's a long distance. There's still over 1,000 bases. And so these viruses are clearly the most closely related to SARS-CoV-2, but they're not the ancestors. They're not the ancestors. Nobody has an ancestor. MERS coronavirus and the human coronaviruses, NL63 and 2290, also came from bats as well. So bats are really important. They have a big diversity of coronaviruses. Two genera in the coronavirus. So the coronavirus family is divided into genera, the genus, right? Genus species. So they're alpha, beta, delta, gamma. The alpha and beta coronas have been found in bats pretty much all over the world. So bats are really important for that. And China has they say China has a rich bat fauna, which means there are a lot of different kinds of bats all throughout China.
Mm-hmm.
Over 100 bat species. And so they looked at sequences of field collected samples from bats across China. And when you do this sampling, you have to get the bat most of the time. And then you can figure out what species it is. Right? So they have that information. And they use that data to answer the questions that we had talked about. So they have 589 partial sequences of the RNA-dependent RNA polymerase from bat rectal swabs collected in China. So when you do a bat rectal swab, you know what kind of bat it is because you're holding it in your hand.
Right.
Okay.
And they also say that they take like a little sample from the wing so that they can do more careful actual identification of the bats.
Yeah, probably mitochondrial DNA or something like that, right? Yeah. Now, you could go into a cave and just collect guano, which is deposited by bats, but I don't think you could know. There are going to be so many different kinds of bats.
It's going to be a pool.
It would be a mess. All right, so 589 partial sequences from bat rectal swabs from China, and they added to that 608 bat coronavirus and 8 pangolin coronavirus sequences that are available in public databases. And then they made 2 databases, one with all the bat coronavirus sequences with the known host, and then one with all the sequences that have a known sampling location. And they used 2 of these, and they divide China into 6 what they call zoogeographic regions by clustering provinces with similar mammalian diversity. They have the southwestern region, the northern region, the central region, the southern region, and Hainan Island. There's a map in here. I think it's open
very
access.
helpful.
So you can.
Both these are open.
It's because they're Nature Communications. Yeah.
Yeah, you can look at the map. China's a big country.
Huge. It doesn't cover everything, but
No, it doesn't.
a large swath of.
Are those gray parts?
Not
those
only
gray
that,
parts
but my guess is it's pretty. I don't know much of the climate in China, but I would assume that the northern region is pretty cold.
Yes,
Probably
it's
fewer
all over
bats.
the place.
And if you look at the and they comment on this in the paper, the bat population, the sampling increases in frequency as you move south.
Yeah.
Among other things, because there's more bats.
Right.
Right. It's colder in the north. And so the gray parts, there's a large section of provinces where they just don't have any coronavirus sequences.
Yep. Okay, so they can use these databases to reconstruct what they think is they make phylogenetic trees, and you can see the ancestral node in the tree, and you can use the bat family as the ancestral character state, and
Wait, as the character state, which what does that mean?
Well, it's the property that they're basing the phylogenetic tree on, right? The bat host family.
Okay. So the tree is organized based on which bats they came from.
Yeah. You could organize it by bat, of course, and you can also organize it by virus. But what they do here is they do alpha and beta coronaviruses separately, according to bat species. And so this suggests that these viruses, for the alpha coronaviruses, originated within rhinolophid and vespertilionid bats. So rhinolophid, the horseshoe bats, and vespertilionid, different species of bats. And this includes HKU2 and SARS-CoV. So HKU2, a human common cold coronavirus, and SARS, the pig diarrhea coronavirus. Others, there's 7 alpha coronavirus lineages associated with a single bat family, but each has sequences from other bat families as well in it. So there's already, they see frequent cross-species transmission events among bats of what is basically the same virus. They're going to look at that in more detail later. So the beta coronavirus is probably originated from not Rhinolophus, but vespertilionid and pteroptid bats. So pteropus bats are the flying foxes, right? The big guys. And they can construct very clear lineages, like the Mirbecovirus, including MERS-related coronaviruses, HKU4 and 5, which are human common cold viruses. And then they have a lineage originating from pteropid bats, which would be like HKU9 coronavirus. So they then use software to look at host switches, transmission between bat families and genera that occurred on their trees. And they have some nice diagrams of circles where each circle is a different species, and they have arrows going back and forth, and you can see they have 9 highly supported interfamily host switches for alpha coronaviruses and 3 for beta coronaviruses. And interestingly, the interfamily host switching is 7 times higher in the alphas than the betas. They don't know why that is so, but more switching in the alpha. I mean, they say in the discussion maybe it has to do with receptors being more ubiquitous in bats for the alphas than beta, but we don't really know what's going on there. Okay, spatiotemporal dynamics. That means the movement across the country with time, right? Spatiotemporal dynamics. And so they identified dispersal routes, and there's another map where they have the different parts of China. Where is it? Yeah, that's figure
6, right?
6. And they have the 6 regions that they've identified, and they have arrows which indicate dispersal routes. So, for example, the Southwest, viruses that they find in the Southwest, they also find in the NO region, the northern region. And there are other regions in between, and they can document all of this movement. They say there are 12 dispersal routes for alpha and 5 for beta coronaviruses within China.
Alpha gets around better than beta.
It does.
Well, it was first. Is it based on the time series of the samples, or how do they know the direction of the dispersal? Sequence similarity?
Probably
from an ancestor?
By the tree, yeah.
Yeah, from the phylogenetic tree, yeah. Yeah. If one is older than another, then you imply that it was right in one place before the other.
Mm-hmm.
So there's one lineage that includes, and then they can put this together with the phylogenetic information and suggest where the viruses originated, right? So the HKU2 and the SADS coronaviruses, they say likely originated in the SO region, while all other alpha coronas arose in southwest China and spread to other regions before dispersal events from SO and NO in all directions. And the SO was also the origin of the Mirbecoviruses, including MERS and other HKUs. Now, this is very interesting. The estimated rate of migration events per unit of time along these routes was 4 times higher for alpha than beta.
Mm-hmm.
So alpha gets around and it recombines, it switches hosts also, which may be connected, right? Because it gets around really well. Maybe it's good at switching hosts.
Maybe the bats also, the Rhinophilus bats also have a wider range.
Let's see. Do they have that on the map? No, they don't.
I don't remember seeing that.
It's a
They
question.
probably said it, though.
I may have said it. One thing that's interesting is that the bat species doesn't seem to play a role in virus diversity, evolutionary diversity.
Mm-hmm.
Right? It's independent of that. So then they wanted to know the hotspots of coronavirus phylogenetic diversity in China. I can't remember what the result was. I didn't highlight it. I didn't think it was so important. But what buried within this is this idea of bat richness. So richness being number of species. So Shannon richness is the number of species, right, Jolene, in bacteria? Is that right?
It is one measure of
One
richness.
measure.
Yeah.
So in bats, you also use the term richness to talk about the number of species. And they say, no, bat richness is not a correlate of coronavirus diversity. Although for alpha coronaviruses, but they did find it for beta coronaviruses. So a significant correlation observed between sampled bat species richness and beta corona diversity at the province level. Okay. Now I'm going to summarize most of the rest because this is a tough paper to read, I have to say. And they don't summarize each section for you. These findings mean this. They don't say that. But in the discussion, they do. So they say, okay, so this work shows a high diversity of coronaviruses from bats. Interestingly, most bat genera in this study, so they looked at 17 different bat genera, 11 of them were infected with both alpha and beta coronaviruses. That's really amazing, right?
Yeah, it is actually.
And I didn't go into this data, but they found that SARS-CoV-2 is likely derived from viruses originating in Rhinolophus bats. I mentioned that at the beginning. And the origin appears to be Yunnan province, but there are a lot of caveats to their conclusion. So they say we can't rule out that these viruses actually came from outside China because they're closely related relatives in Myanmar, Laos, Vietnam, and maybe elsewhere that we haven't sampled. So Yunnan is close to the border of these countries, right? So it could be Yunnan or it could be some of the other countries as well.
Right.
And we've talked about some sampling studies like in Laos, where they find very closely the banal coronaviruses, very closely related viruses. They also have some isolates from pangolins here. Everyone probably knows that they were able to isolate SARS-CoV-2-like viruses from pangolins, but they say that their analysis of sequences say that the bats are more likely to be the origin of SARS-CoV-2 than pangolins. Okay, so then, of course, the other conclusion is that there's a lot of cross-species transmission over evolutionary time. What kind of time are we talking about? Well, Rhinolophus, they can find fossils of Rhinolophus bats in China, which are 38 to 48 million years old. So they've been around a long time. And in fact, maybe they originated in China and then spread eastward in Asia and eventually to Europe. And so if that is where a lot of these viruses originated, they are quite old. And these bat species, the Rhinolophidae, have played a key role in the evolution of these viruses. Now, these horseshoe bats, share roosts with many, many other kinds of bats. And that's probably one factor in having the high cross-species transmission. What did they say about the Rhinolophus bats are the biggest donors and the biggest receptors in cross-species transmission? Something like that. I don't remember exactly what. Where it was. But that's what they basically said. Host phylogeny doesn't have a significant. It does. There is an effect of the phylogeny on beta coronavirus evolution, and minimal on alpha coronavirus diversity. Some bats were infected by a single genus of coronavirus, as opposed to many bats being multiply infected, and they don't know why that would be the case. So they say that the southwestern and southern China are the centers of diversification for alpha and beta coronaviruses, and other viruses too. Probably originated there, like influenza viruses H5N6, H7N9, H5N1. So they say this is where we should be sampling. Sampling and discovery should target these two hotspots in southern and southwestern China and neighboring countries, which don't get sampled very often. So these regions are unique. They have a subtropical to tropical climate. They're dense, rapidly urbanizing populations of people. They have a lot of poultry and livestock. All this can promote cross-species transmission. So we should really know what's in these regions. And as they mentioned, SARS-CoV and SARS-CoV both emerged in this region.
I thought it was, well, I had to look up this word. They said, oh no, now I lost it, that this region, southwestern China, acted as a refugium during Quaternary glaciation for numerous plant and animal species, including bats and other mammals and so forth.
Yep.
So, I mean, because of the density of species that radiate from there, it makes an ideal environment for that sort of.
Yep. And now you throw people into the mix, right? And you can get cross-species, you can get spillovers into humans, and that's the concern. So they say we should be discovering viruses in China and across Southeast Asia, southwestern China. And the bat species, Rhinolophus, but also Hipposideros, Myotis, and Minioteras are all involved in switches throughout coronavirus evolution. We should target those for discovery. So this is very cool stuff, and it's what we need to be doing. Now, as Rich posted an article in here, EcoHealth Alliance lost their NIH grant, right?
Twice. In 2020, when the lab leak thing first made the news, and they threw EcoHealth Alliance under the bus.
Yep.
Because they represented a collaboration between the U.S. and the Chinese to figure out where these viruses were coming from. And then more recently, this article talks about that in 2024, I think, actually.
Yeah.
They eventually regained their funding, but then they lost it again. The grants were closely looked at by a congressional committee, and they found some what they judged to be, say, sloppy grants management, and some evidence that some of the work in China, we've talked about this before, there was some work that here would have been done at BSL-3 that there was apparently done at BSL-2. And they didn't like the whole thing, so they cut them off again. So you can read that article. One of the interesting things is that if you read the acknowledgments, in the acknowledgments for any given paper, they'll say what their funding sources are.
Mm-hmm.
And it says here that it's funded by the NIH and from a USAID grant. Says all work conducted by EcoHealth Alliance staff after April 24th, 2020, and that's when the funding was cut off, was supported by generous funding from, and they name a number of donors here, which are basically private donations. Including
Literally anonymous donor care of a charitable foundation.
Right. So,
And
you can look at this how you will, but the work is important, and to have it really needs this kind of global collaboration. All right. So I hope in the long run this kind of thing continues.
I think also in the spirit of scientists doing the best science that they can. This article is newly published, but the study was done previously and published. And they found an error in some of the original data sets, as in they had included some that they didn't mean to, that weren't sampled from China. And so they redid the whole analysis and got it re-reviewed and published again.
So
I think that, Rich, they're working to get it back again, their grant.
oh yeah, it'll all work out. If we don't blow ourselves up first. None
of the complaints really justified taking the grant away.
No,
the administration
I don't
stuff.
think so.
There were a few progress reports that were late. So what?
Yeah,
Who
right.
really cares? The other thing is they were doing it at BSL too. Okay, that's done. That did not lead to the pandemic. Okay, right. We know this. So it's done. Make them do it at BSL 3 if you want, but to take it away is ridiculous.
It is, shall we say, tainted by politics.
Yeah, and unfortunately, some prominent scientists have testified in front of Congress that this was bad that they were doing BSL 2. So it gets messed up.
So, but the bottom line for me is that these bats, and there are plenty of them, are crawling with these viruses, and they're passing them back and forth, which in my mind, as I think there's potential there to accelerate the evolution. I'm kind of making that up, but when you cross a species, there must be some adaptation that goes on, I would assume.
Yes,
sure. And so that means that's going to contribute to the diversity. And as we've already said, it focuses attention on a particular region that historically, because of its richness in virus sequence diversity and in bat populations, has apparently given rise to numerous spillovers over time into humans. And so it's an area that ought to be watched. And it will be interesting to see whether in the long run that geography can be expanded to include the neighboring countries in an even larger collaboration.
Well, I think the borders they're not ecological borders, right?
No, that's right. As a matter of fact, they make that early on when they talk about their geographic study. They say we chose to define our own geographies rather than using political boundaries, because the political boundaries don't make any sense.
Right.
Okay. But there are definite, geologic boundaries. Yeah, the bats don't know about the political boundaries.
No, and it could have been any country, but it happened to be that it's China, and it's got this ecology, and it leads to a lot of viruses in bats, a lot of coronaviruses. So we just need to deal with it. You can't be accusatory, right? So
it is a fact.
It is.
These are facts.
These are facts. All right. Now we're going to move on to something totally different, but still a virus, right, Jolie?
Still viruses. That's right. So I'm going to talk about a paper also published in Nature Communications earlier this year, 2024, called Diverse and Abundant Phages Exploit Conjugative Plasmids. There are co-first authors on this paper, graduate student Natalia Quinones Olvera and former postdoc Sharon Owen, as well as Lucy McCulley, Maximilian Marin, Eleanor Rand, Alice Fan, Oluremi Dosumu, Kay Paul, Cleotilde Sanchez Castaño, Rachel Petherbridge, Jillian Paul, and it comes from the lab of Michael Boehm. And they are at Harvard Medical School. So this is the paper itself to give kind of the overview of what we're talking about here. I'd say that here, really, the researchers who pull viruses from the environment noticed that using classical techniques that we love here on TWiV called plaque assays, we were getting the same thing over and over. And so what they did was they added an extra layer of information to the plaque assay. And this extra layer of information allowed them to pick out more rare viruses when sampling from the environment. And so that's great because these rarer phages have a lot of new genes in them that could become interesting new biotechnology tools. And the extra layer of information that they added cuts down on a lot of the tedious labor that you would otherwise have to go through to find those rarer phages from the environment.
Plus, the techniques are cool.
Yes, the techniques are very cool. And the kinds of phages that they are finding in the past, these kinds of phage, which are dependent on the plasmids that they're talking about, have been things like the ones that were the first viruses who had their genome sequenced, and the ones that were the source of the technology for phage display, which won the Nobel Prize a few years ago. So finding viruses with new genes and kind of new functions can lead to a lot of cool new biotechnology. And so to be a little more specific here, all phage, just like all viruses, have to interact with some aspect of the surface of the host cell to gain entry. And a lot of the time, the phages that we study are interacting with membrane proteins on the bacterial cell surface or decorating sugars. And these are usually encoded by things that come from the chromosome of the bacteria. But there are also phages who interact with molecules or assemblies of molecules on the surface that are encoded by plasmids. And one example of that are pili, which are bacterial.
Let's make sure that the folks out there know what a plasmid is.
Okay, yes. A plasmid is a circular piece of DNA smaller than the chromosome. And they're actually, sometimes they can be linear, but usually they're circular. And so they replicate independently of the chromosome. They can be single or higher than single copy in the cell. And they're tools that we use for doing all sorts of manipulations of cells.
And they can do things like carry genes that are important for the bacterium, like drug resistance. Okay.
Yeah.
And they can also move by conjugation from one bacterium to another. So that's bacterial sex, where two bacteria interact and one passes DNA to the other. Quite often just the plasmid, and the plasmid actually can code for the apparatus that does that conjugation, called often called a pilus, and code for the machinery that does the transfer as well. These plasmids are this is DNA wars. Okay, the plasmids don't want other plasmids around, so they have genes that make it so that the environment is hostile for another plasmid. So they become incompatible with one another. And they are grouped as such. Am I getting this right, Jolene?
Yeah, that's right.
So you have incompatibility groups, which is where this nomenclature Inc. Inc. and a letter come from. Inc. P, Inc. F, etc.
Right. Yeah, so plasmids can encode antibiotic resistance genes, and frequently that's how they're passed around between bacteria. And they can encode these appendages called pili. Sometimes they're conjugative pili or type 4 pili, which can be involved in surface sensing, motility, or uptake of things like DNA from the environment.
And in fact, the phage we were talking about before, M13, was that during recording or before we started?
That was before we started. That we ought to, we don't do Easter eggs anymore. That whole pre-show conversation was great.
But those M13s attached to pili, right?
Right.
Interesting.
So the phage that attach to pili, there are several that are known, but not very many. And we call these plasmid-dependent phages. We always thought that plasmid-dependent phages were relatively rare, but here in this paper, they're going to show us otherwise. So what they started with was knowing that there weren't very many of these plasmid-dependent phages. There had been some reports of them back in the 1970s and 80s, where there were at least 39 different plasmid-dependent phages that were reported that targeted different kinds of plasmids that were in the different incompatibility groups. But those phages are no longer available for study. They were lost. And so we really don't have very many that we can culture and study in the lab anymore. So what they did here was they took a really clever approach. So basically, the phage requires the presence of the plasmid because if the plasmid isn't there, it can't make the pilus that the phage has to bind to get into the cell. So it can only grow and make plaques in a plaque assay when the plasmid is there. So to isolate more of these plasmid-dependent phages, they put these colorimetric markers or fluorescent proteins encoded in different species of bacteria. And another interesting thing about these plasmid-dependent phages is that plasmids tend to have a broad host range, meaning that they can be present in bacteria of not just different species, but different genera. So here they are working on some plasmids that can be found in Salmonella enterica and Pseudomonas putida.
Macroevolution.
Yes, more than one different kind of, not just different kind of species, different genera.
Wow.
And Salmonella enterica and Pseudomonas putida at the start of this study didn't really have any phages that were known to infect both of them, but they could grow well together. And they did know that there were certain phage that could infect plasmids that would be found or that could be cultivated in Salmonella and Pseudomonas. So they picked a couple of these incompatibility plasmids, an INC-P plasmid and INC-F, and they put fluorescent tags in each of these bacteria. And then they would mix the two bacteria together. And so if you just had one bacteria and you plate it in a monolayer on a plate and let it grow up, it makes by eye, it makes an opaque layer of growth. And by imaging the fluorescent protein, it makes a colored layer. And then the other bacteria will make the other colored layer. So when you mix them together, they just look like this kind of yellowish opaque layer, but you can separately image the two fluorescent proteins in different channels and then merge those together. And then when you mix phage into that co-culture of these two hosts that make different colored lawns, then image them, what you get is the opaque layer of growth, where you can't tell which is which, but then the fluorescent layers where you can see which host has grown. And then you have the plaques. And so the plaques are usually clear spots or clearings in the lawn. So you can kind of see through it and it looks darker. And if you look at the colored channels, any phage that could infect both of the hosts will form a completely clear plaque because it killed both of the hosts. But if it could only infect one host, then the other host will have grown and that'll be the color of the plaque. So this co-culturing method allows them to differentiate between phages that can infect one host, the other host, or both. And they call this the plasmid-dependent phages by fluorescence-assisted co-culture, or phage disco, which is a great name. So they used this phage disco method to screen several environmental samples to look for phage that could infect both hosts when the plasmid was present. And they were looking at samples that came from wastewater, as well as, I think there were a couple of other samples. Right, compost and farm waste, all from the greater Boston area, which is where they were located. And they had conjugative plasmids, INC-P and INC-F. And then they after establishing that this could work with Pseudomonas putida and Salmonella enterica, they switched the disco host strains to E. coli and Salmonella because that allowed them to try different plasmids. And they also had good genetic backgrounds for getting rid of other plasmids that way. So they collected from this co-culture, this disco co-culture method, 50 phage that were dependent on the INC-P plasmid. So if INC-P was there, they could make a plaque. If INC-P wasn't there, they couldn't make a plaque. And then 13 that were dependent on the INC-F plasmid. And they also did the screening without the presence of the not in the co-culture, in the monocultures, and they found some that just infected those that required a specific host to be able to infect.
So I'm gonna go back here a little bit because when I was reading this, I was thinking about this selection differently.
Okay.
So the idea is we want out of this phages. It's a quick way to distinguish between phages that can infect one or another of these specifically, or both, right? And they're focusing on those that can infect both,
yes,
right?
Because the plasmid is there.
Because the plasmid is there. Now, the assumption there is that they're different plasmids, right? But the assumption is that they have something, they have a number of characteristics or encoded functions in common, presumably. So the phage is using those two plasmids in the same way.
So they did the screen using INC-P in both hosts or using INC-F in both hosts.
I see. Okay. I'm sorry. I thought it was one or the other. I had it wrong. Okay, fine.
That's fine.
Okay. That makes sense then. So you're distinguishing guys that distinguishing ones that are plasmid specific from those that are host specific. Got it.
Exactly. Yep.
By the way, this is open access again, and the figure one where they describe this is gorgeous.
Yeah, and they show their very clear demonstration of this working when you have a phage that can only infect one or the other based on the presence of the plasmid versus a phage that can infect both. So they isolated 65 total plasmid-dependent phages from this screen, and they gave them all names that, of course, based on the theme, had colors in them. And so this naming system that they used was according to how similar the new phages were to previously known phages, and then they added a color to the name. So they got lots of interesting names like PRD chartreuse or PRD avocado, PRD peacock, cobalt, pistachio.
PRD cerulean.
Cerulean. That's one of my favorite words. Cerulean.
Oh, really?
Yeah, I like it.
Just because it sounds cool?
Sounds cool. Yeah. It's a good color, too.
I actually didn't look up what color it is.
Yeah. Look it up. You see. Better than me describing it.
Okay. So then they went about classifying them. And so that was part of the naming. So there were INC-P plasmids dependent phage that turn out to be similar to TWiV viruses. Now, TWiV viruses are really interesting. So TWiV viruses, they are phage, and they're phage that have lipid layer on the inside of the capsid. And they are DNA, right? Wait, no. They're RNA. Now I'm confusing myself.
I think they're. I thought they were DNA.
Hang on. They are DNA.
Yeah, I looked it up.
Double-stranded DNA. Okay. Double-stranded DNA. This is in the first figure, actually. But with a lipid layer on the inside, which makes them like very interesting. And there were not very many alpha TWiV viruses known previously. So they show us that there were these essentially 6 previously known alpha viruses, but then they searched the literature, and based on genome similarity, there were 7, because there was this other one from Burkholderia. So the classic well-studied one is PRD1. And so that's where they got the base for the names of the rest of the ones that they named. And they show that a small percentage of them are very similar to all of the known alpha tectes, but the rest of them were quite different. They're estimating that based on the way that we classify viruses with nucleotide similarity, we've gone from just having 2 known species to 12. Which is quite a lot. And they also note that the previously isolated tectiviruses came from a huge geographic range of places on like 1, 2, 3, 4, at least 4 different continents. But their sample came from just the Boston area, and they found a huge number of new alpha tectes, which suggests that these are greatly undersampled in the environment, and that there are probably a lot more alpha tectes out there than we ever knew before. And then they also looked at the places that these were similar to the ones that were already known across the entire genome. So, we know that they're alpha tectes based on similarity. They have a similar genome organization, but the actual nucleotides all along the way are different. Just like we saw with those coronaviruses, there are certain regions of the genome where there's more differences. And so they saw a couple of hot spots, but actually the differences were spread all the way across the whole genome among the set. And then they looked at the next set, which were single-stranded RNA phages, and these are dependent on the INC-F plasmid. So the INC-F plasmid, it had the single-stranded RNA phages that they compared to were Q beta, MS2, and PRR1. So these are quite well known, or have had a lot of studies done on them previously. So MS2 and Q beta are part of the Phages-Viridae, now known as Phages-Viridae single-stranded RNA phages, and PRR1 was a little bit more removed than the others. However, they show that their single-stranded RNA phages are similar to all of these, but they also kind of cluster separately. Meaning that not only have they found phage that are closely related, but also phage that are not closely related in their sampling. And they had a set of filamentous-like phages that were dependent on INC-F. So these are enoviruses. They have actually single-stranded DNA as their genome that is packaged. So the one that we were talking about that is the single-stranded DNA phage that was used for phage display and for many other applications is M13. So there are some of the examples of what they found there were FF amethyst, FF lilac, FF iris. So these filamentous phage are some similar to M13, but some very different again. And then the last one that I'll highlight here is the INC-P-dependent phage that is actually a double-stranded DNA phage that is tailed. And so usually when we do these isolations from the environment, we find tailed phages, and they have double-stranded DNA genomes. But here they only found 1 out of all of the 65. And so that was a little bit of a surprise that it would come out in this kind of a screen, but interesting because it is dependent on the INC-F plasmid. There are a few other tailed phages known to be dependent on pili, but the way that they interact with the pili isn't really well understood. So this one, they called it FT Midnight, and it has a long flexible tail, And they imaged it so they can see that. And they also were looking at the similarities between the tail genes of FT Midnight and other phage tail genes. And so they kind of think they've narrowed down which of the tail proteins is going to be important for interaction with the pilus.
I would guess it hits the end of the pilus, right? Because otherwise you'd have, if it hit the side, the DNA would have to go in at a right angle. Probably not happening, right?
Well, so the way that usually for the tailed phage, the DNA is going to come through the tail, but the proteins that interact with the receptor are not at the end of the tail, if that makes any sense. Yeah. So if you think about the tail is like a central tube, and then the fibers that typically contact the cell are away from the tube on long extensions. And so the actual angle of genome delivery is not going to be dependent initially on the contact with the cell surface, because it can reorient itself.
This one, Midnight, doesn't have any tail fibers, though, right?
Well,
not that we can see on this picture.
It doesn't look like it has long tail fibers, but that doesn't mean that it doesn't have any tail fibers.
All right, maybe it has whiskers.
That's possible. We do call them whiskers, but
yeah, I know. When they're shorter, right?
Yes, and also actually usually closer to the neck.
Right.
Yeah. So, right. So with the FT Midnight, they found part of how they were doing the analysis of which proteins might be involved in the binding to the host cell. They looked for some colonies that could grow in the presence of the phage. So they basically found resistant mutants. And these resistant mutants of the host still had the plasmid, because it still had the antibiotic resistance marker. And these resistant mutants were also resistant to MS2 and Q beta, which are two single-stranded RNA phages that bind to the pilus. And those bind to the side of the pilus, not the tip, actually. And so this suggested that there was a particular part of the machinery involved in conjugation that was important for FT Midnight to be able to infect. And so, let's see. Okay, so that's kind of an overall description. Using the phage disco method, they could screen the environment, and they found 65 new phage, all related to previously isolated phage, but not very closely related. There was a large amount of diversity coming from a small geographical area. So another thing that they were interested in is, if they were able to find these plasmid-dependent phage, then could they learn something about why these phage had a particular host range? Or maybe they all could infect any bacteria that had the plasmid that they were dependent on present. So they took a large panel of bacteria, including the originally used Salmonella enterica and E. coli, but a bunch of other ones as well. And those included things like, I'm looking for the list here. Things like, I don't know if I can find the short names. Okay, I'm gonna go based off of what I remember. So Klebsiella pneumoniae, there's a Citrobacter in there. I think there is an Enterobacter, some Pseudomonas. And so what they did was they mixed together the growing culture of each of these bacteria containing the plasmid, and then they infected each of them with the phage that they had isolated. All the TECD viruses in this case, so PRD, asparagus, pine, mamacita, azure, indigo, etc. And they looked at the growth patterns of the host. So they looked at plaques and growth patterns. So in plaques, they could get an idea of how big are the plaques, how many plaques could they make on one host versus another. We call that efficiency of plaquing. And with the liquid growth assay, they could see, does the host still grow? Does it grow at the same rate? Does it eventually lyse? So they assigned this score based on the growth patterns, and then they plotted those. And they see that some phage, though not very many, are really good at infecting almost any bacterial species that they tested. But most of them are really good at infecting a couple and not really good at infecting the others, even though it has the receptor. So that suggested to them that the phage isn't just dependent on the presence of the receptor, which is not really a surprise. It's just there aren't very many cases where we can measure something like this, because most of the time the receptor isn't so easily moved from one host to another. So it's actually, I think, really interesting that the majority of them seem to be not completely restricted, but have preferences, let's say, on which hosts they're going to grow really well in and which ones they're going to plaque really well in.
It also means to me that when you do this screen, you're going to pick up only a subset of
That's right.
all of the phages out there that might interact with a given plasmid, right?
Depending on which bacteria you're using as the host for the plasmid. Exactly. So it varied a lot, but there were a few that were of interest, some that really could infect almost everything. And so one possibility for why this was that there's different physiology in the different hosts, or it could be that the different hosts had defenses that prevented the phage from replicating really well in those cells. So they did look for the presence of defenses, and those weren't really found. They didn't seem to have phylogenetic relationships that could predict those preferences, and they also looked at the sequences of the CRISPR-Cas and restriction modifications found in the host genomes, and they didn't all have the same kinds of defenses. So there wasn't a strong correlation there. So it really seems to be pointing to, although there's not a lot of evidence for it yet, that there's differences in the physiology of those host cells that will be dictating how well the phage can replicate. And then they did something that I thought was really exciting, and that's they looked at the PRD cerulean phage, which could, if you look at the range of the hosts that it could infect, it really only did well on one host, essentially one. And so they took that and they used a high titer to try to force it to infect others, and they passaged it until they could get a spontaneous plaque former on one of the other hosts. And then when they sequenced it, they found that it had mutations in a lysis gene. And this lysis gene turned out to be the holin protein. So the holin protein is the initiator of the lysis process at the end of the infection cycle when the phage is going to be released into the environment. This is a membrane protein, and it accumulates in the inner membrane of gram-negative bacteria, which is all of what they're studying here, until a certain time, and then it forms these giant holes or small holes, and those holes kill the cell and simultaneously release an enzyme that can degrade the cell wall, and then eventually the phage will get out after that. Those physical barriers are gone. And so these mutants that they found were all in the holin protein, or the gene for the holin protein. And they found a few of them. So the mutations weren't always in the same spot, but they were in the same gene. They also had a couple of other mutations in the genome, but those were not as correlated with this phenotype of being able to replicate in both Salmonella enterica and Pseudomonas putida. So the wild type cerulean only replicated in Pseudomonas putida, but all of the Holin mutants could also form plaques in Salmonella enterica. And they saw that these mutations were in the transmembrane regions, which is something that we've seen in the past as a place where mutations in that protein can change the timing of the phage exit, but we haven't really seen it associated with host range before. So that's really interesting. Unclear at the moment how the amino acid sequence in the membrane would have an impact on the ability of the Holin to work. Because we've traditionally thought that the Holin is a master regulator of timing, but it should work in any membrane because we thought that it was protein-protein interactions between Holin subunits that were necessary for the formation of the whole and initiation of lysis. But here it seems like something else in the membrane, perhaps protein components or perhaps something about the lipids, is going to be really important, and changes in the Holin protein allow it to work in the other host cell now. So then they did some swaps of these genes. They swapped out the mutated genes and put that into the wild type, and they put in the PRD1 into the PRD cerulean, and they showed that it was really the Holin protein that was allowing it to expand its host range. Maybe there were also other things, but the Holin protein on its own was sufficient. Now, returning to this question of they found a lot of new things, but why haven't they been found before? Mm-hmm. So they looked at what I thought was this really interesting. So I've never thought about, because I didn't really have a way to measure it previously, how many plaque-forming units are present in raw environmental samples. So I've mostly done phage hunting with E. coli. If you take wastewater samples directly from the environment, no filtering, no anything, and you just put it directly onto a plate, then you will see plaque-forming units. And here they measured how many plaque-forming units of the alpha TWiV could they find in their different samples that they had. And for the ones that were plasmid dependent, those seemed to be about 1,000 PFU per ml. 1,000 PFU per ml. And for E. coli, they tell us that there's about 4,000 PFU per ml, E. coli specific phage, as then they also measure it for Salmonella, it's about 100 PFU per ml. And then for Putida, it's 10 or less. And so that's quite high. Therefore, you would think that if you sequenced this sample, you would find the alpha TWiV sequences. So they looked at the sequences. Well, they did sequencing of the samples, and they looked at the sequences, and they didn't really recover very many alpha TWiV sequences in their sequencing run. They also looked at the metagenomes from many other publicly available data sets, and the abundance of alpha TWiVs in those were also quite low. So anywhere between, say, 0 to 5 reads, and based on the abundance that they measured in their sample, there should have been more. Now, why are there not so many reads in the sample? Unclear. They also looked for assemblies. Assemblies are definitely not there, but raw reads, they say maybe it's because there's a low abundance of the phage in the environment, or because the genome is small. So the alpha TWiV genome is about 15 kb or less, and many of the double-stranded DNA phages are 40 kb and up. Or perhaps the highly polymorphic population is responsible, they say, for this. I'm not really sure that those are clear explanations for why they're not in the raw reads. I'm wondering about the contribution of the techniques used for generating the libraries, whether that has something to do with excluding these samples. We know, and they say this, that sometimes there are proteins that are covalently conjugated on the ends of the DNA, and that can inhibit the formation of the libraries that are used for sequencing in these metagenomic data sets. So maybe that's also part of it.
Yeah, when you, depending on how you make your DNA, I'm reminded of the discovery of the terminal protein in adenovirus that I think made it so that it did not extract into the aqueous phase as well when you did a phenol extraction. I don't think I'm making that up. Okay, so you could have some selectivity against a virus that had a terminal protein.
Right.
Yeah, which they mention here, right?
Yes. Yeah.
This is so it's adenoviruses, right? They mentioned potential precursors to adenoviruses, or at least that mechanism of a protein priming mechanism,
right,
for genome replication. Adenoviruses and these phage have in common.
Yeah. Also, possible that some of the enzymes that are used are biased against these DNAs, and that might be part of it as well.
So, Lean, if they went back to their original samples and just redid their screen, would they still be pulling out the same viruses they identified, or do you think they would get some new ones?
You mean with the co-culture in the same hosts?
Yeah.
You know, I did not see them talk about how many times they isolated the same phage.
Good point.
So possible that they would isolate new. I would expect some repeats.
But I'll bet you put the same plasmid in different hosts, you'd get a whole new set.
Yeah. I'm just wondering how saturated the screen was, that's all, using the same hosts.
Yeah. I don't know, but they got a lot of new phage. If they really only had a few samples, it seems like there's quite a few to be isolated. So I think, to kind of recap for this study, they added fluorescent proteins to specific bacterial species and then mixed them in co-culture. And then they used that as the infection lawn for screening environmental samples. And then they could look at the color of the plaques to determine which host was susceptible or not. And if you put into these hosts a plasmid that contains the receptor, only the one with the receptor will die, and so the other one will make a color. And this method allowed them to isolate the plasmid-specific phages that we hadn't seen before and hadn't been able to culture. Now they are culturable and isolatable from the environment without having to go through the labor of finding phage that are the ones that are similar to what we've already seen before. And this allows us to have new kinds of phage for study. And they also mention that I think is really important to think about is that they can expand this technique. And so they actually just they just deposited a preprint in bioRxiv last month. One of the authors here on this paper, Ellie Rand, is the first author on the preprint, where they show that they can use this phage disco method for screening different things. So they put on it's basically whatever's on the plasmid. So if you put on the plasmid, say, a phage defense system, and then you screen your phages, then you can look for the ones that are sensitive to or resistant to the phage defense system on the plasmid. And they show that they can mix more than 2 colors. So they have hosts where they have 3 colors and more. And they show that they get these kind of intermediate plaque phenotypes where the plaque will be completely clear, but they'll have a halo that's just 1 color. And that's kind of based on what you put on the plasmid and how important that is for the ability of the phage to replicate and the cells to die. And I think that's really great. The only caveat is that it has to be genetically tractable. So you have to have hosts where you can put in these colors. But then after that, it's very simple to do the phage disco method. And then you also get these very colorful plates that you can just be happy looking at.
What other bacterial species could you use besides the ones they use here?
Well, a lot. Even though it's not easy to put DNA into many different bacteria species, there are many, many gut bacteria, soil bacteria that we can put plasmids into. So you just have to be able to get DNA in. I don't know, to name like a few, there would be, they talked about Burkholderia. I know you can do Burkholderia, many different kinds of Pseudomonas, Enterobacter. Let's see. What other kind of, they show us there are some environmental bacteria, Klebsiella, Serratia, Proteus, Mirabilis. I think this one is a Shigella. So I'd say that
There's a lot.
there are many.
Yeah.
Not everything, but many. And with a lot of the tools that we find a lot of the tools we develop by finding new phage, we increase the genetic tractability of bacteria that were previously not genetically tractable. So that number will only go up.
I think it's really cool that this stuff is going on.
Yep.
You know, phage still rock. It's great.
They also disco.
They also disco.
Yeah, apparently they disco. Let's see where the funding for this.
Some of it's from NIH.
Oh, yeah.
National Institutes of General Medical Sciences.
Yeah.
Mm-hmm.
Where the hell is this? It's all at the very,
end.
It is. Yeah, actually, there's a lot. NIGMS, the David and Lucille Packard Foundation, Pew Charitable Trust, Alfred Sloan Foundation,
NSF.
and NSF.
NSF is a very typical place to get phage money from, right? Because, you know, the NIH says, well, it doesn't have anything to do with human health, so you go to NSF. I remember when I first wrote grants when I went to Columbia, I sent it to NIH, and he said, this is too human health related for us. It was a poliovirus grant, I think.
And at NSF?
NSF, yeah.
I see.
NSF got me launched.
Really?
Because I you know, at the time you could submit basically identical grants. Not basically, identical grants.
Really?
It was routine to submit the identical grant to NIH, NSF, and American Cancer Society. So my first grant went to all three agencies, and all three agencies blew me away. And then I got this call from a program director at NSF. Said, you know, we got a little extra cash here. I understand that you've been, you know, you're having trouble getting this rolling. So we'll give you half the money for half the time. And I said, works for me.
You know, that's back in the day when these program people actually cared about people's careers.
Yeah, and the program people at NSF, I think, had some more latitude than
that's true.
Some of the other agencies.
So I see you put in a image of MS2 binding the F.
Yeah, because I remembered having seen images of that before.
Okay,
this
so it's a 90-degree. It's a 90-degree angle, so no problem in getting DNA at a 90-degree.
Well,
angle.
this is MS2, so that's an RNA phage.
Yeah, actually, this is not the latest structure of it, but it's not quite a 90-degree angle. It's a little more. It's because there's the maturation protein, which actually binds to the pilus, is at an angle.
I see.
And it's different for Q beta and several other RNA phages, but similar concept in terms of where the binding occurs on the side of the pilus.
Listeners, this is the dangers of anthropomorphizing. You know, I just felt that a 90-degree angle would be too much for DNA based on human quality. You know, if I was building something and something was going to come, I wouldn't do it at a 90. So it's dumb, you see. Biology does not pay attention to human quality.
Yeah, but then you have a discussion with your science buddies, and your science buddies discourage you from going off on that anthropomorphic ride.
The interesting thing is we really don't know how the RNA phage gets its RNA into the cell. Like, we don't really think it's necessarily going through the pilus, and it doesn't have its own like ejection apparatus. So
So is the pilus actually like a tube that during conjugation DNA goes through?
So There is evidence suggesting that the DNA goes through the pilus. There's also some thought that it doesn't really go through the pilus. It's still an active area of research. I would say, since it's not my area, I don't want to commit to one place or another, but I've seen some really cool videos showing two bacteria next to each other, pilus connecting them, and DNA going pretty much where the pilus is from one cell to another.
But not necessarily inside?
It's very hard to show.
Hard to tell.
Very hard.
Okay.
Thanks, Jolene.
Yeah.
Cool.
Yeah, I
Very
think
cool.
this is a really cool story, and I know that some of it, the work is being followed up on in the BAME lab, and that Sharon has started her own lab at the Wadsworth Center and is taking students as well. So great things to come from here. If you want a disco, talk to these people.
Let's do a couple of emails. Rich, can you take that first one?
Sure. Mike writes, from Denny, either Dure or Dure on Instagram. If you were evil and wanted to destroy America, how would you do it? 1. Divide society. Encourage division across political, racial, and ideological lines, amplifying conflicts that create distrust and weaken national unity. 2. Weaken education. Gradually lower standards and shift focus away from critical thinking, creating a population that is more easily influenced and less informed. 3. Damage public health. Promote widespread use of unhealthy foods, chemicals, and pharmaceuticals, leading to chronic illness and dependency. 4. Break community bonds, undermine family and community structures by fostering isolation and individuality over collective support. 5. Control the narrative. Limit free speech and censor alternative viewpoints, creating a culture where people fear speaking openly. This would erode resilience, leaving the nation open to influence and control. What's happening in America is not an accident. It's part of a plan. As a Cold War veteran, I recognize the Soviet template. The USSR may be gone, but their work marches on. TWiV is part of the solution. Shine a light on ignorance and refute lies and misinformation. You've inspired me to do what I can. After the election, I was so demoralized that I gave up for a while. If the forces of truth and knowledge surrender in this battle, the country loses. Mike. So to me, the two words that are the most important in that whole thing is critical thinking.
Yep.
That's, in my mind, that's, if I have a mission in TWiV, it's that. Demonstrate critical thinking.
And don't give up.
You know, the you see on the news all the time, all of this stuff about what can we do to stop the spread of misinformation and disinformation, okay? Talking about, identifying it, making sure it's, not published or amplified, identifying sources, blah, blah, blah. But it's kind of like drugs. The really thing you need to do is to treat it on the consumer's end. Because in the end, it's up to the people to whom this kind of stuff is directed to be able to discriminate between what's real and what's not. And that's what we need to do.
Yeah. Well, Mike, thanks for your service. And I think that, as we're reading this list, it is a little discouraging if you think about it like that. But the part that we can control, what we do, who we talk to, what we talk about, those are the places where, you can't give up. We still need you.
Right. Sounds like Mike still has some energy. That's good.
Great.
Jolene, can you take the next one, please?
Yep. Chris writes, "Good morning, Earth's virologist." To be read like "Good morning, Vietnam," which unfortunately I do not know the reference. So I can't get the right intonation.
That's, I'll do it for you. That's Robin Williams. Good morning, Earth's virologist.
Very good.
Thank you. You've made my brain sizzle again while listening to TWiV 1175 about worms carrying viruses and the billions of people carrying them. You asked at time 5445, what's going on here? And a bit later, speaking of the worms living in the viruses, it seems to me they are okay with them. This cuts both ways. Humans are okay with the worms, and the worms are okay with their viruses. I think we never eradicate anything because it would take too much energy and work. Our bodies attack invaders and beat them down to a point where their interference doesn't matter enough to worry about. We have better things to do. We always live with bugs and their bugs if they don't actually bug us. We always don't want to get rid of anything that can actually help us in some way. Our evolution is often the result of making use of their complexities to make beneficial mutations. I guess it's truer to say that whatever doesn't kill us might make us stronger. As the guide says, our bodies don't panic just because there are bugs on board. It's the same as the Vogons allowing the Dentrosi to be on board their spacecraft because they're the best cooks in the universe, even though they pick up hitchhikers. But Ford and Arthur were not helpful by rejecting Vogon poetry and got thrown into the void. I wonder what Dixon would say about this. Are we unable to eradicate all our bugs, or is it evolutionarily advantageous not to? Happy holidays and New Year to all our hosts and helpers and to everyone lucky enough to listen. Fondest, Chris Way, still hitchhiking on your ship, Toronto 34 Fahrenheit with gray socked in clouds.
That's very nice. Thank you.
This is quite insightful here. I think the notion that we beat them down until they don't bug us anymore is good. It's not necessarily eradicating them. And, that's probably, the origins of a lot of symbioses and et cetera. Pretty good. And these must all be references to Hitchhiker's Guide to the Galaxy?
Universe. Is it Galaxy or Universe?
Hitchhiker's Guide to the Galaxy. I need to go read that again.
Okay, one more from Charles. Hello, Twivers. It has turned cold in Chapel Hill, North Carolina. It's just 36F2C as I write this after a wet day. In previous TWIVs, you have pointed out Trump's unqualified people to lead public health. I have not heard you mention Dr. Jeanette Neshiwat. When I first read about her, she seemed out of line with the rest of Trump's picks. Then I read some more. I am not a fan of people attributing to God all good things and not holding God accountable for the bad. Her book, Beyond the Stethoscope, bugs me more than a bit. What upsets me is she's trying to sell me B plus C boost for $22.94 a month to support my immune system. This puts her in the Senator Orrin Hatch snake oil sales tent. What do you think? Because of her New York location, do you have any firsthand experience with her? Thanks, Charles. That's Dr. Oz, too. He's selling snake oil. Well, he doesn't sell it, probably. He recommends it. I don't know Dr. Neshiwat at all.
She's got a Wikipedia page here.
I was going to write a video, Charles, just like the others, but I don't know why I didn't. But these are the points that bug me also. Mostly, there's some things about COVID that she said that were not right. And then that she's making her own supplements, right, to sell to people, which is ridiculous, right? So I think this is a problem. Now, the interesting thing is that she, so she's the medical director of CityMD, which is a chain of places here. Right, there's one right down the street here. You can go in if something's wrong with you, right? Minor stuff, and they can help you. And they're owned by United Healthcare, which owns Daniel's organization out there as well. So, but he doesn't know her. He's being very politically correct these days, so he wouldn't tell me what he thought. But I think she's in the group with the rest of them. I mean, they're all unqualified, in my opinion, right? And for various reasons. So this one is no better. No, I mean, they all have the veneer of being qualified, right? They're MDs or they're from Stanford or or Johns Hopkins, but you just look a little deeper and you realize they're not. And, the problem is that you can not nominate any one of them, but they'll just come up with another wacko who's just as bad. You're never going to get a good person for this. This is very bad. Very, very bad. Okay. Yeah, we have more about this. Just Charles has a long letter about Jay Bhattacharya, which we'll read another time. Right now, we're going to do some picks of the week. Rich, what do you have for us?
So I stumbled across this while we were recording last week, and it's from a kid's book or a book for kids in the Berkeley Library, and the book is called "The Discipline of Organizing" for kids, and it's all about how to organize stuff. Apparently, there's a book written for adults about organizing stuff, and this is for kids. But this is an activity in the book. So I just picked this one page describing a phenomenon that I didn't know there was a word for, called pareidolia, if I'm pronouncing that correctly, which is the tendency to see in various objects recognizable, in particular, human characteristic images. And the one they show here is 6 different images that all look like faces, and they're hilarious.
Yeah.
Top of a coffee mug with, a happy face in the foam, and the standard, the 3-prong receptacles. I love the dryer.
Yeah.
Okay. With the clothes hanging out looking like a tongue.
Exactly.
So I got interested in this phenomenon, whatever it is. You can, it actually comes from the book so that you can click over the contents and you can see about the whole book. The book is online. And I also looked it up, as I always do, on Wikipedia. And there's a Wikipedia link there, too, that describes the phenomenon. It's the tendency for perception to impose meaningful interpretations on a nebulous stimulus, usually visible. And then they have all sorts of scientific explanations for why this is and what areas of your brain get turned on when you do this, and et cetera. But I just think it's interesting that we want to see faces and stuff. Isn't that interesting?
Yep.
I can't explain it, but it's interesting. And these are for sure faces.
I mean, even every individual image in that picture, I would definitely see the face.
Oh, absolutely.
That's so cool. Jolene, what do you have for us?
I picked something called pathwaystoscience.org, which is a database from the Institute for Broadening Participation. And this is an organization that has compiled a series of opportunities for trainees at many different levels, including summer research opportunities and funding availability for undergraduates and graduate students, so that they have for other levels of scientists as well. And I think this is a place that I send students very often. Especially at this time of year, I'm suggesting to them, and they're thinking about what are they going to do next summer. And oftentimes I'll send them to the National Science Foundation REU search database so that they can look for places to apply. But the Pathways to Science database includes summer research opportunities, but lots of other things, internships and programs from different schools and departments. And I think it's just a really great compilation of things. So
cool.
Yeah, this is a resource especially for students looking for things to apply for.
You might be able to not even wash dishes somewhere. Oh, by the way, in the phage article, if you looked at the acknowledgments, let me do this here. I've got it right here. We also thank the other instructors and students of HMS Phages 2022 summer course. And they have a bunch of names. which was partially supported by HSMI Science Education Alliance. So this is, I guess, one of, I don't know if this is Phage Hunters or another similar program that the Howard Hughes Institute does, where kids can, look for phage in the dirt.
Yeah. It's
Yeah.
they've
Cool.
extended to do different things.
Hmm.
So definitely there were undergraduates involved.
It's great.
I think some high school students too.
Yeah.
Good. All right. I have a pick. I don't know where I was trying. So I was trying to find this album this week. Vinyl. I own a lot of vinyl, and for some reason, one called "Switched On Bach" came into my head. Now, I had bought this album when I was an undergraduate at Cornell. When did I go to Cornell? In 1970. So probably was 70, 71 or so I bought. Because I'd heard about this synthesizer. And Robert Moog, who developed the Moog synthesizer, was at Cornell. And so I couldn't find the vinyl. It's just hard to find. But I started looking into, I remembered that Wendy Carlos had composed the album. So I looked it up. And so this pick is her Wikipedia site. And it's really a fascinating story. So she went to Columbia to study music composition, got into electronic music, which was just being developed at the time. And she helped Moog develop the synthesizer, right? The first electronic instrument. And the first iterations that he built were these huge boxes with cables that you had with jacks on the end that you had to connect to. So there was no keyboard in the earliest iterations. You made sounds by putting cables into different places. So he had a series of oscillators that made different kinds of waves for sound, and then you could change. His revolution was he made it so the voltage would change the pitch of the wave, and so then you could control it that way. So Wendy Carlos got fascinated by this, and she got an early prototype, and she decided to record a bunch of songs by Bach, right? So she did it. Every note had to be recorded individually. You would plug cables in a certain way. You get the note, and you record it. And she recorded everything on an 8-track recorder. Remember those things? Actually, no. That's an 8-track is a substantial record. You can do 8 individual tracks. And she eventually made 2 sides of the LP. What is this? Tells you here how long it took for her. I think it took her 8,000 hours to record this. She had somebody help her do it. So the Moog could play one note at a time. Due to the limitations of her equipment, every instrumental part in each score had to be recorded separately, and then each part was layered over the previous recorded part on her 8-track recorder. All parts were coordinated by recording each one with a click track, which eventually was erased. The biographer, her biographer, reports that she rarely wrote down the combination of control knob settings and cable patches for making sounds. She could just sit down and do it, right? But she never wrote it down.
Wow.
Boy, you got to really want to do it.
So it took 1,100 hours to complete. She had a full-time job on top of it. So this was released in 1968. It was a smash hit. It was the first album made with the Moog synthesizer.
Mm.
It hit platinum, the first classical album to ever hit platinum. It got 3 Grammy Awards. So platinum is like, I don't know, it's over 1 million copies. 1 million copies is gold, I think. I don't know what platinum is. But it was just a huge hit and nobody expected it. But it introduced the synthesizer to the world, and musicians of the world suddenly took note. And the album is fantastic. I played it all the time, and I can't find it. I want to find it and play it.
Mm.
So she did another album called the Well-Tempered Synthesizer, which is the Well-Tempered Clavier is I think it's a piece by Bach, right? So that's cool. And then Kubrick had her do the score for Clockwork Orange and another one of his movies. What the hell was it? Clockwork Orange and The Shining. So she did a lot of stuff. And so basically this album was a turning point in music, right?
Mm.
And I was in a record store in Ithaca in 1971, and I'd heard about this synthesizer, and I'm looking through, and I see this thing switched on box. So I just bought it randomly. I had no idea what it was. And I remember the first time I played it, it was just mind-blowing. It's just so good because it's all these Bach pieces and totally electronic, right? There's no instrument at all. So this kicked Moog into doing other things. So he eventually made what's called a mini Moog, which had a keyboard. So then you could put it on stage and musicians could play it. And one musician said, this now makes a piano equal to a guitar. Because, the guitar ruled rock bands because you could do so much with the guitar and pedals, effects pedals. And, a piano couldn't do very much. But now this thing, you could play anything. And in fact, for a while, it was outlawed because they thought it would put session musicians out of business, right? Because this thing would, say, play a violin, but that's not how it works. It's a totally different sound. So now everybody's using these things in their productions, and music that you hear probably has some synthesizer in it as well. So I just thought this story was really interesting. I tried to find it online. They never licensed it for streaming or digital download. So you can only buy the LP or the CD used. It's out of print. You cannot find it online, which is really interesting. And so Wendy is still alive. She's up in Rhode Island. She's 85 years old. And she has a website, wendycarlos.com. I just think it's such a cool story, right? And these people just experimenting with stuff and coming up with amazing, amazing advantage. That's the way science works, right?
There are a couple of YouTube clips here, but I guess.
So there are YouTubers people have taken the LP and they've played some pieces from it, but it's crappy because they don't do a good job. You're not allowed to redistribute it. It's not licensed. So
I think Bach would be pleased because he was
Glenn
nuts.
Glenn Gould said this was the album of the decade, and Glenn Gould was a very controversial pianist. But he was all about trying different things, right? And so this obviously tweaked his interest. But I remember this album. It has on the cover, it's a picture of a guy who's supposed to be Bach sitting on a bench, and behind him is this huge synthesizer with cables snaking out of it, right? He's got a pair of headphones on his lap. So, he's got the white curly hair and the shiny satin clothes and it's just and there are very few notes. You don't know anything about it, right? Some LP notes are huge, right? They explain all about it, but this one had nothing, and I got to find it, and so I can play because I loved it. It was so cool. And the second one I never bought, so the well-tempered synthesizer. That sounds cool. All right, so I just thought that.
Yeah,
that was
very
really
cool.
awesome.
I like that.
Nice.
Actually, it reminds me of. I mean, I thought it was kind of a riff on Alan's pick from last week, where you had a band that was playing electronic music. Okay, I don't know if that was last week. I think it was last week because I got off on that and listened to a bunch of that stuff. It's very good.
Oh, was that the German band?
Yeah.
Oh, yeah, it was a listener pick. Yeah.
A listener pick. Okay.
Yeah. They were playing. What were they, like a marching band playing?
Yeah, there was an 11-piece band, a lot of percussion, and a bunch of horns
Right.
that were playing, instrumented versions of electronic music. And it was wild. Great stuff.
Cool. Yeah, you said you were going to go listen to it right away.
I did.
All right, we have two listener picks. Arjan writes, Dear Team TWiV, since you have a more than passing interest in human evolution, may I suggest Gutsick Gibbon's YouTube channel? Gutsick Gibbon, a.k.a. Erica, is a PhD candidate in biological anthropology. On her channel, you may find videos concerning primates, general zoology, paleontology, anthropology, and evolutionary biology, as well as a hefty amount of time debunking young Earth creationism. Her videos are very informative and are presented with a pleasant sense of humor. So if you're a gentle and, of course, very modern ape, this is a must-see channel.
Cool.
It's great. Sounds great. YouTube is really cool. You can find a lot of neat stuff on it. I mean, you can find a lot of wasteful stuff, but you can find such amazing stuff that people do like this, or people make stuff, and it's amazing.
And it just goes to show you how just unimaginative I am. Because when YouTube first started up, I thought to myself, what?
What? Yeah, I know. I did too. And when Google bought it for $1 billion from this kid, I said, what? But that's the thing. You got to have vision and foresight. You got to think that something is going to be useful and work. I have to check this out. This sounds great. Thank you, Arjun. And Alan writes, if you haven't caught up with the 2024 Nobel lectures, you'll be fascinated by the lectures from David Baker, Demis Hassabis, and John Jumper. Tons of virological and immunological implications and the birth of a new generations of tool. And it gives a link to the lecture. These are the AlphaFold winners.
Okay.
Yeah, AI. Consider it a listener pick. All the best, Alan. Thank you, Alan. That'll do it for TWiV 1181. You can find the show notes at microbe.tv/TWiV. You can send emails, questions, comments.
Bags of money.
Whatever you want.
Gold.
Picks to TWiV at microbe.tv. If you want to send us some support, some financial support, you go to microbe.tv/contribute. You know, we have these autographed copies of Principles of Virology, autographed by all 5 authors. And this year during our fundraiser, anyone who gave $1,000 or more is entitled to one. I just shipped one off today. But many people have given us $1,000 or more. You know, you can get your textbook. You just have to tell us you want it.
Until supplies run out. But we'll tell you after you give your $1,000.
Yes.
Just kidding.
I
Just kidding.
think I have 9 copies left. So I don't know if we have 9 people who have given that much, but if you want it, it's a cool thing to have. And I'm happy to get them out of my house because they take up a lot of room. Put them in yours instead. Microbe.tv/contribute. Jolene Ramsey is at Texas A&M University. RamseyLab.Vercel.app. Thank you, Jolene.
Thanks.
Rich Condit is an emeritus professor at the University of Florida, Gainesville. He is currently in Austin, Texas. Thank you, Rich.
Sure thing. Always a good time.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. I should switch the music to Switched on Bach, right, for this episode. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.